being able to identify what stain you're looking at, especially after midterm, you guys are going to get your own unknown organism and you're going to do all these stains again, right, to identify your unknown organism. So it's important to know what staining procedure you're doing, to be able to look at it on the microscope and determine what was done or potentially done, especially if you don't label your slides good, right? So we started out with very simple stainings, simple stain and negative stains. And because cells themselves have an overall what charge? Negative. They have an overall negative charge. They respond differently to the different charge dyes we use. So basic dyes have what charge? Positive, right? So some of the ones that we've used are crystal violet, methylene blue, and saffronin. We'll use some of these again today. As you see in the picture, because they're basic, they're positively charged, they stain what? The cell, right? They're attracted to those negative components, opposites attract, and the cell gets stained. Now for negative staining, we use an acidic dye. Acidic dyes have what charge? Negative charge, right? They give up their protons, they become negative. So some examples of some acidic dyes we've used in this lab are Congo red and nigrazine, and then mentioned in one of your homework assignments was the eosin. We don't actually use that one in this lab. So again, they're negatively charged. So cell's negative, dye is negative, what happens? They repel each other and the background gets stained, right? So the cell actually repels the dye, so the cell isn't stained, but the background is stained. In this picture, what shape is the individual cell? It's a rod, right? What do we call this chain of rods? Streptobacillus. The individual cell shape is what? Coccyx, right? And there's a set of 4 in this square, tetrad. For the gram stain, right? First you start with a heat-fixed emulsion. Most of the stains that we do, you're going to start with a heat-fixed emulsion. Right? There's just 2 that we do not. Which 2?
Negative.
Negative staining.
And
starts out like a negative stain. Did it last week. Capsule stain. Capsule stain, right? Your negative stains, and your capsule stain starts out like a negative stain, right? But then you stain additionally. And this was the reason, remember last week, why we didn't rinse it off. We just blotted it dry because we didn't heat fix that one. Because heat fixing does what? It shrinks them, but the reason why we do it is to stick them to the slide, right? To stick the bacteria to the slide so that when we're staining, especially stains that we're going to rinse off, we don't rinse off our cells. It kills them, it shrinks them, but those are byproducts of why we do it. Right? What I was really looking for on your quiz was adhering to the slide. Okay, so once you have that heat-fixed emulsion, for the gram stain, what's the first dye? Crystal violet. All the cells get stained purple, right?
yes.
Then we rinse with deionized water. What's the next step?
Iodine.
Iodine. This binds to the crystal violet and makes it a bigger molecule, right? Then rinse after a minute. Then we decolorize with what?
Alcohol.
Ethanol, which is an alcohol. Either is acceptable. You actually use 95% ethanol. Dropwise until it just starts to lose its color, right? What happens at this step? Gram-positive cells are going to stay purple. Why?
Because their membrane is...
Not their membrane.
Their cell wall.
The peptidoglycan cell wall does what? Shrinks and traps in the crystal violet iodine. For the gram negatives, they have peptidoglycan, but not enough to trap it in. And then they also have another layer that gets removed by the ethanol or seriously damaged. What layer is that? The outer membrane, right? So at that point, the gram negatives are colorless, right? So in order to stain them, you must counter stain them with safranin. Rinse, blot dry, oil immersion. What magnification is oil immersion? Total magnification?
100 times.
100 is the objective, 10 is your ocular, so total magnification, 1000, right? We put oil on it to help with the refractive problem of light. If there's a big gap and the oil is not filling that gap, you're not in focus. Always with bacteria, right? We go all the way to oil immersion, 1,000 times magnified. Even then, right? They're teeny tiny, right? You guys looked at those microscopes, most of them. So for the gram stain, you have two possible outcomes if performed correctly, right? For the first one, what color are these cells? They're purple. What stain made them this color?
Crystal violet.
Crystal violet. And through the process, they stayed purple, so therefore they are gram positive. Remember, this is a result. This is not having anything to do with charges. These cells are what color?
Pink.
Pink. What stain made them that color?
Safranin.
The safranin. And they are gram
Negative.
negative. Both these pictures show what shape of cells?
Tetrads.
This has tetrads. This one, I see a few tetrads. I see more pairs, though. See more pairs. But definitely cocci, too, right? Probably diplococci for this one. If you were to scan around and look, you'd probably see more pairs than anything else. And when you're trying to determine arrangement, the most complex arrangement that you see the most often is usually what you have for that organism. Remember, cocci form lots of arrangements, and we'll go over those. So if we had the ability, which we don't, right, but the gram stain gives us clues into this on how they respond to the procedure and the steps of the procedure. The gram positives, which tend to have the very thick peptidoglycan layer, right, that trap it in, these guys only have one membrane, the cell's membrane, right, which has multiple names in the literature, right? Cytoplasmic membrane, plasma membrane. I'll always refer to it just as a cell membrane. But know that when they say these other terms, they're meaning the same thing, right? Just the cell's membrane. And for gram positives, their peptidoglycan layer, this special substance that makes up the cell wall of bacteria, is very thick. In contrast, gram negatives have how many membranes? Two. The cell membrane, and then what's the other one called? The outer membrane. That's pretty easy, right? It's the outermost covering. Now, chemical composition, that one's actually quite different than most membranes. Their peptidoglycan layer is thin, and that's the problem, right? It can't trap in the crystal violet iodine. So this is a capsule stain example. If I didn't tell you, how would you know that this was a capsule stain? You see the white halo, so you know that's a capsule. But what about this picture? Is there a capsule? No, A is the one that has the capsule. But how do you know that's a capsule stain?
Because we stain the background.
And the cell, right? The cell and the background are stained, right? If just the background stained, then it's just a negative stain, and you are not going to be able to detect the capsule. Right? So picture A has the capsule, and for capsule staining, you've got to stain the background and the cell in order to see the unstained capsule, which is outside of the cell. So outside the cell wall, or outside the cell wall and the outer membrane, if the cell has it. What are the two functions of the capsule? Protection from phagocytosis, right, from being destroyed by our white blood cells in our body, and adherence, being able to adhere to attach to surfaces like our body. Acid-fast staining you're going to do today. This is the color, right, that you'll see. It's kind of a purplish pink, right? In one of the videos they actually said hot pink. I was like, that's a new term. But I agree. It's kind of a bright kind of pink, or sometimes referred to as fuchsia. What stain, what dye are we going to use today is going to make them that color? We're going to use carbofusion. And we're going to use a particular formula of carbofusion, the ZN formula, because we're going to use the ZN, the Zielen-Neeson method, for acid-fast staining. If they come out, they stay this primary stain color, we say that they're acid-fast. We don't, like in the gram stain, usually we say gram positive and gram negative. For this, we say acid fast or non-acid fast. But you could say acid fast positive and acid fast negative. But traditionally, we say acid fast or non-acid fast. Now, notice that these cells clump together, right? But when you see the individual cells, they are rod-shaped, right? And that's why I was so excited about my slide today. You could actually see individual cells, right? And you could see that they were rod-shaped. So a common abbreviation that you'll see is AFB, which stands for bacillus, right? Not bacteria, bacillus, right? Although they are bacteria, and in fact of a genus of bacteria called mycobacteria, right? That we'll talk about in the lecture for today. So other cells will stain whatever dye you're using as a contrast, as a counter stain. So these cells are kind of a bluish, grayish blue. I'm not sure exactly. I stole the picture from the internet, you know. It met my criteria. And again, it depends on which dye you're using. So the protocol we're using today calls for methylene blue. If you're using the Kenyon method, typically they would use brilliant green, which doesn't necessarily look beautifully green. We've used that one in the past. They look kind of gray if you ask me. And then in the videos, if you watch the videos that I had for you guys, do you guys remember what different counter stain they used? Crystal violet. They used crystal violet. You can use whatever you want as long as you can tell the difference between that and the carbofusion. So I wouldn't suggest using like saffron, right? That may be difficult to determine the difference. If you deviate from protocol, what should you do? Record it, right? Write it down. Right, so I had one group last class, you know, they're like, oh, let's use the crystal violet just for fun. Let's have some purple cells. So that's fine, but for testing purposes, right, they're going to look like my slide. They're going to be a bluish color. But they're definitely not going to be the characteristic color of acid fast. So therefore, they would be non-acid fast or acid fast negative. Basically anything other than, right, that hot pink fuchsia color. The other stain we're going to do today is the endospore stain. And again, there's lots of different methods out there. The method that we use is the Schaefer-Fulton method. So for that one, our endospores will be what color? Green. And I don't know about you, did those look green to you over there? They look bluish to me. They look like aqua, maybe. And so even, I don't know, I guess that looks green. On here it looks not completely green. I don't know what it is with our particular malachite green, which is the dye we use. It seems more of a bluish color to me. So it's another reason why I have that demo slide up for you guys. Plus you'll see them when you stain them today, right? That they, I don't know what it is about the malachite green. In the past it was like Christmas tree green. It's more blue lately. But again, it's going to be within that color range, right? Greenish, bluish color. So as you can see, the cell in this picture has the endospore within it, and that's why it's called an endospore. It forms within the cell. Then eventually the cell would deteriorate. As you saw in my slide, there was a lot of what we call free spores, right? You had to kind of look around to see the red cells and potentially see some inside. So on the test, under the microscope, I'm not going to ask you arrangement like you see here. As you can tell, it's really difficult to determine. So it'll be more like pictures like you had on your quiz today. But this is a really good picture. You can really see really well, right, the arrangement. So which one of the terms describes the arrangement of this spore within the cell? Subterminal. Do y'all see that? It's not smack in the middle, which would be what? Central. And it's not at the end, which would be terminal. This is in between the middle and the end, right? So it's subterminal. Do you see any spores in this other picture? No. No. Just cells, yeah. There might be like an artifact, but if you were to see spores, you would see them, right? And again, you would make sure you scan around on your slides, right? You might not, you know, there's lots of surface on that slide. You're only looking at a small portion under. But in this case, no green structures, no endospores. What conditions are necessary for an organism to produce endospores? Harsh conditions, right? The one that we employ very easily in the microbiology lab is starvation or nutrient depletion. So although usually, typically in this lab, we work with relatively fresh cultures that are 1 or 2 days old, these cultures now are about 4 days old. So they've pretty much run out of nutrients. And so they definitely have produced spores. And we did that on purpose, right? Because we want you guys to be able to stain spores today. So stain identification. So what do you see? You see colors, right, which will help indicate you, and you see colors in particular places. So this one's an endospore stain, right? You see the endospores within, right? You see the cells. And it's that red and greenish color. The next one, what do you see stained? What colors do you see? The background and the cell are stained, right? So it's a capsule stain, but does this organism have a capsule? No, because there's no clear halo, right? There's nothing unstained in between the cell and the background. The next picture, what colors do you see? What indicators do you see that would let you know what staining procedure was used to make this slide?
No background.
Right, so we're just staining the cells, right? We've got that purplish pink color, don't we? And the cells are clumping together, but you can see in some places where they're kind of rod-shaped. So that's a pretty good indication that this is probably an acid-fast stain. And it is. This one you see purple and pink, so that one's our gram stain. And this one, what do you see?
Negative stain.
Just the background stain, right? So it's a negative stain. You'll see all 5 of these, right, on your test next week. In addition, when you look at these, you can tell other things about the organisms, right? We can see shapes, we can see arrangements here. So you got to know all your shapes and arrangements, right? So A is coccus if you're talking about one. If you're talking about all of them in that square, cocci. The next one, diplobacillus. Diplococci. I'm all right. If you're talking about all of them, diplococcus if just one. What's the arrangement in the next picture? Tetrads. Next picture. Strepto, right? This is the chain. The chain is strepto. Remember strep throat. Think about how you would want to swallow a chain. The next one is a packet of 8 cells. This is Sarcinia. And then the clustering, Staphylococcus. These guys, the term, Bacillus. And then a lot of times we refer to these as what?
Rod-shaped.
Rod-shaped, right? The term for this next arrangement? Diplobacillus, or Diplobacilli, if you're talking about all of them. And then the next one, Streptobacillus. And this one is a rod that has a little bit of a curve to it, right? This is Vibrio, and it also shows what? It has flagella in this picture. The next one is a longer rod with more than one curve to it. Spirillum, or spirillia, if you're talking about more than one. And then if the organism does not hold a characteristic shape, being pleomorphic or pleomorphism. And then these guys look like ziggly lines under the microscope. These are your spirochetes, or spirochete. And these guys have that ability to corkscrew through their environment. So for midterm, all the exercises we've done so far, including today, your quizzes are a good example of the type of questions I will ask on the exam. Right? So review those. You should know all the terms you've been assigned for homework. This includes spelling them, as I said. There will be lots of slides you will have to identify, both the eukaryotes, right, which students aren't so fond of. The good news is they're only on the midterm, they're not on the final. Unlike bacterial smears, right, that'll be the whole semester. You still had a question? Yeah, just the genus names. Just the genus names, but that includes the identifications, right, and questions. And remember, I still have those pictures, that practice quiz you guys can do on Canvas for the identification of the genus, right, the first name for each one of those organisms. But you'll be looking at them under the microscope. So, again, like that quiz I had kind of set up for you guys in the bonus one that you should be able to still see your results for. This would be how it would probably be set up a station, right, where I would have these questions. I'd have the aspergillus slide there. You'd have to identify it's aspergillus, that the pointer is pointing to the asexual spores, right? And I could ask the question about what commercial product this is produced by this organism, right? So make sure you review all the stuff about those guys. You'll need to know all the steps to all the staining procedures and why you see what you do, right? And the staining ID assignment that I just went over is available to you online, both with the answers and without, right? So you can kind of self-quiz yourself. And then I have a bonus quiz for you guys, and then also an actual quiz 6 that has more questions. It's nothing but stain identification. More of the results than actually identifying the stain. I actually tell you what the stain is. I probably should add some in the future of what is this stain. Okay. You should be able to distinguish gram-positive from gram-negative, right? Acid-fast from non-acid-fast. You should know shapes and arrangements and things like that. And so those additional resources will help make sure that you guys know that. We've used and will use all of these organisms on this list, and we've used them for particular reasons. Mycobacteria sphegmatis is what's going to be on your slides for you today. That's your acid-fast organism. Its shape and arrangement is that it's bacillus, right? It's rod-shaped, and it tends to clump together. Not to say that, well, I don't know. I guess the cells probably even stay together, but they're not. It's not quite like the arrangements that you see with other organisms, and that's to do with this type of organism, this special type, this acid-fast organism. E. coli, this one is rod-shaped. Does it have an arrangement? No, it's singular. They don't really stay together in particular arrangements. If you did the stain on it, it would come out pink. So therefore it is gram negative. I have acid fast on here to really drive home the point that the only one that's acid fast on this list is the first one. It's mycobacteria. All the rest are going to be what? If you were to stain them, non-acid fast or acid fast negative. Micrococcus luteus. You guys remember the shape of this one?
Spherical.
Yeah, spherical cocci, right? It's in its name. This one forms that cool packet of 4 cells, so it's called what? Tetrad. Gram stain, it would come out purple, so it's gram positive, and then acid fast would be non-acid fast, right? Or acid fast negative. It's not a mycobacteria, is it? So it would be non-acid fast. Staphylococcus epididymis, which is the one that you're going to have today, is your non-acid fast, right? It's not going to stain with the carbofusion and stay stained with it. It's going to be whatever your contrasting color is. If you were to do the gram stain for this one, it would come out purple. So it's gram positive. I forgot I have these written in. It's cocci, right? Staphylococcus, its name itself lets you know that it's cocci in clusters, right? Klebsiella pneumoniae. Anyone know why, what special structure this one has, why we chose to work with this organism? You're thinking of mycoplasma. It causes pneumonia, right? Why does it make us sick? What is it that's special about this bacteria that contributes to it making us have pneumonia and infection of our lungs? A capsule. It has a capsule. It sticks to us and avoids phagocytosis, one of our defenses of our immune system, right? And so therefore it can cause a really serious form of pneumonia. So that's why we had chose to work with that one, right? Its shape is rod-shaped. It doesn't form a characteristic arrangement, so we refer to it as being single. It would be gram-negative if you stained it. All right, the main reason why we used it is because it produces a nice, thick capsule. Not to say that some of these others don't have capsules, right? Do you guys remember which one, Micrococcus luteus or Staphylococcus epididymis, which one had a capsule? The staph did, right? The Micrococcus luteus does not. Although really hard to see under the microscope, huh? Because these cocci are pretty tiny to begin with. Bacillus, as the name implies, right, what shape are these? Rod-shaped. And most of them form chains, so that would be Streptobacillus. If you were to gram stain them, they would come out purple, so they're gram positive, and all of them would be for the acid fast stain? Non-acid fast. And then the reason why we're using these today is what special structure do these bacilli produce when they're starved? Endospores, right? And so each table will have a different species of bacillus that's an endospore former. So make sure you indicate which one your group has on your guys' sheets today. So remember, the gram stain doesn't just tell us positive and negative. That positive and negative tells us about the cell wall structure, right? So for gram positives, we know that's a very thick layer of the peptidoglycan, that special substance that makes up the cell wall of bacteria only. And then for the gram negatives, that means that they have a thin layer, and they probably also have that outer membrane.